Okay, let's go ahead. Okay, so plants and other photosynthetic organisms contain organelles called chloroplasts. You've heard of these before, probably in your middle school or high school biology classes, so nothing new here, and you're probably a little bit familiar with photosynthesis, but it's the process that converts solar energy into chemical energy within the chloroplasts. Directly or indirectly, photosynthesis nourishes almost the entire living world. Remember, this is pretty much talking about the creation of the bottom of the food chain. Autotrophs. Auto means cell, so these are cell feeders that sustain themselves without eating anything derived from other organisms. They are the producers of the biosphere. They produce organic molecules from carbon dioxide and other inorganic molecules. Almost all plants are photoautotrophs. They photo being light reactive, so photoautotrophs using the energy of sunlight to make these organic molecules. A picture of a tree, and so again, everything that goes into producing the organic and inorganic molecules are necessary for this tree to survive or produce or as come as a direct result of sunlight. So photosynthesis occurs in plants, algae, certain other unicellular eukaryotes, and some prokaryotes. Here's some different examples. Your standard plants, you have algae and unicellular eukaryotes in the bottom left, cyanobacteria top right, and then other things like purple sulfur bacteria. Now, heterotrophs obtain organic material from other organisms. They are the consumers of the biosphere. Some eat other living organisms. Others, called decomposers, consume dead organic material or feces. Almost all heterotrophs, including humans, depend on photoautotrophs for food and oxygen. So the Earth's supply of fossil fuels was formed from the remains of organisms that died hundreds of millions of years ago, hence the name fossil fuel. Fossil fuels are being consumed faster than they are being replenished, and researchers are exploring methods of using the photosynthetic process to produce alternative fuels, as you'll see in this next diagram here. Okay, chloroplasts are structurally similar to and likely evolved from photosynthetic bacteria. Their structural organization of the organelles allows for the chemical reactions of photosynthesis to occur. Leaves are the major locations where this happens, and chloroplasts are found mainly in the mesophyll, which is the interior tissue of the leaf. Each mesophyll cell contains 30 to 40 chloroplasts, and carbon dioxide enters and oxygen exits the leaf through microscopic pores called stomata. A chloroplast has an envelope of two membranes surrounding a dense fluid called the stroma. Please keep stomata and stroma clear. They're kind of similar in spelling, but very different structures. Thylakoids are connected sacs in the chloroplast that compose a third membrane system, and the thylakoids are stacked in columns called grana. And chlorophyll, which is a pigment that gives leaves their green color, resides in the thylakoid. So here's a picture here at the top left. You can see they're showing you where the mesophyll is located in the leaf, and they're showing you a cross-section of that leaf. You can see the stomata, which are the pores where the CO2 goes in and the oxygen comes out. You can see the chloroplast inside the cross-section of the leaf. And then they're showing you a mesophyll cell in particular, and you can see inside you have the thylakoids, and the stack of thylakoids are called a granum. The fluid around the granum, kind of like a standard eukaryotic cell has uh, a fluid on the inside. These do too, and in this case it's called the stroma. Okay, and again, that's different than the stomata, which are the pores. So the stroma is the fluid on the inside of the mesophyll cell. And then you have an inner and outer membrane with surrounding the mesophyll cell. Okay, so photosynthesis is a complex series of reactions that can be summarized as the following equation. Carbon dioxide and water in plus some light energy will produce a sugar, oxygen, and water. The overall chemical change during photosynthesis is the reverse of the one that occurs during cellular respiration. These are why these go hand in hand. Photosynthesis produces the sugars, oxygen, and some water, most of which are the inputs into cellular respiration. And out of cellular respiration, you get carbon dioxide production, some water released. and so these things go round and round, they feed each other. So the overall chemical change during photosynthesis is the reverse of the one that occurs during cellular respiration. Now, chloroplasts will split water into hydrogen and oxygen, incorporating the electrons of hydrogen into sugar molecules and releasing oxygen as a byproduct. So this is showing you where, from the reactant side of the equation, carbon dioxide and water going in, where they end up in the products. You can see everything's utilized and it goes someplace. So clearly the carbon dioxide that goes in is the same carbon that gets utilized to make the sugars. Okay, so the carbon dioxide that we exhale as a result of cellular respiration is the input for plants to gain carbon and to convert it and use it to make sugars that we will then later consume and recycle this process. Photosynthesis reverses the direction of electron flow compared to respiration. It's a redox process in which water is oxidized and CO2 is reduced. Now, photosynthesis is an endergonic process. The energy boost is provided by sunlight. So here this is showing in this equation, the carbon dioxide becomes reduced as it forms a sugar, and water is becoming oxidized as it forms oxygen gas. Two stages of photosynthesis. This is a quick overview. You have the light reaction, the photo part, and the Calvin cycle, which is the synthesis part. So you have the light part, the photo part, and the synthesis part. Now in the light reaction, which occurs in the thylakoids, you're going to split water molecules, release oxygen gas, reduce the electron acceptor NADP plus to NADPH, and generate ATP from ADP by photophosphorylation. The Calvin cycle, the second part, which occurs in the stroma, forms sugar from carbon dioxide, uses ATP and NADPH. This Calvin cycle begins with carbon fixation, incorporating CO2 into organic molecules. So here you go. You can see in the light reaction here, you have sunlight coming in, hence the name of the light reaction, and water. You have NADP plus and ADP plus a inorganic phosphate going into the light reaction in the thylakoid, which is floating in the stroma fluid inside the chloroplast. Now, as a result, the ADP and P are going to be joined by photophosphorylation, form ATP. Then NADP plus is going to be reduced by adding the electron from the hydrogen, and then as a result, the water molecule is going to get broken up and produce the oxygen gas, and that's the oxygen that we breathe. This is the O2 that actually leaves through the stomata. Okay, in the Calvin cycle, a CO2 comes in. This is the CO2 that enters into the cell through the stomata, or into the tissue through the stomata, or the cell actually, and then it's going to get carbon fixed in the Calvin cycle and then go through this process. And as you can see, ATP and NADPH are going to feed in, and then they will in turn get broken back down to ADP and an inorganic phosphate and NADP plus to be reutilized again in the light reaction. So for every turn here, while this is turning, it's producing NADP plus and ADP and inorganic phosphates and taking them, putting them back together through the light reaction into these, which will then feed back into the Calvin cycle. Again, your output here is going to be your sugar, and those are the energies that we generally get at the base of the food chain. Okay, chloroplasts are solar-powered chemical factories. The thylakoids transform light energy into the chemical energy of ATP and NADPH. Light is electromagnetic energy. It's also called electromagnetic radiation. It travels in rhythmic waves. Now the wavelengths, the distance between crests of electromagnetic waves, and wavelength determines the type of electromagnetic energy. You may remember all this stuff from a physical science type class or physics in the past, but you probably are a little bit familiar. We'll talk a little bit about it right here just so you have a background of how this applies to the light reaction. So the electromagnetic spectrum is the entire range of electromagnetic radiation or radiation. Visible light consists of wavelengths from 380 nanometers to 750 nanometers. This is the range of wavelengths that we can actually see with the naked eye. Visible light also occurs or includes the wavelengths that drive photosynthesis, and light behaves as though it consists of discrete particles called photons. Now here you can see visible light all fits right here. To the left of our visible light spectrum, we have ultraviolet, and to the right, the red spectrum of light, we have infrared. The two UV and infrared, which we cannot see, but you can also see there's X-rays and gamma rays that fit into here as you get significantly smaller. And the nanometer scale, as you go up in the nanometer scale and get into larger metric-sized wavelengths, you get microwaves and radio waves and such. Okay, so the big thing here with visible light, the shorter the wavelength, the higher the energy. So when you start getting down to the left side here, as you start moving in this direction, things are getting smaller. So as you start moving in this direction, they get smaller and have higher energy, whereas longer wavelengths over here in the red spectrum are lower energy. And you can kind of see this effect if you look at a lighter or something. If you strike your Bic lighter or something like that, you know, the outer color of the flame is always a little bit cooler, and as you get to the inside where you have higher energy or a hotter flame, you can see it's blue. So just a very easy example of taking a look at some of this stuff. But certainly you can see lots of examples of this on YouTube and videos of wavelengths of light and the amount of energy they contain. So pigments are substances that absorb visible light. Different pigments absorb different wavelengths, and wavelengths that are not absorbed are reflected or transmitted. And leaves appear green because chlorophyll reflects and transmits green light. So here you go, you got light striking it, and the wavelengths of light that are not reflected are absorbed and utilized by the thylakoids, and the light that's reflected actually comes back, and that's what we see. So things look green because they're showing us the reflected light off of the organelle, not because it's being absorbed and utilized by the plant itself. So a spectrophotometer measures a pigment's ability to absorb various wavelengths. The machine sends light through pigments and measures the fraction of light transmitted at each wavelength. And the absorption spectrum is the graph that plots the absorbed light versus wavelength. Okay, so here's an example. If you have a white light bulb, an incandescent bulb, you use a refracting prism, and you have a solution of chlorophyll here in both cases. If you adjust the slit so that you only pass the green light through, and you have a photoelectric tube reader here, you'll see that there's high transmittance or low absorption, meaning it's going through, it's not being absorbed, and it indicates that chlorophyll absorbs very little green light. On the other hand, if you adjust the slit so that you pick up a different wavelength of light, in this case the blue light, you'll find that as the light passes through the test tube of chlorophyll, that you'll get very low transmittance, meaning it very little passes through, it gets absorbed, and the reading indicates that chlorophyll absorbs most blue light. Now there are three types of pigments in chloroplasts. There's chlorophyll A, which is the key light capturing pigment, chlorophyll B, which is an accessory pigment, and carotenoids, which are a separate group from chlorophyll B, but they are also accessory pigments. The absorption spectrum of chlorophyll A suggests that violet, blue, and red light work best for photosynthesis. An action spectrum profiles the relative effectiveness of different wavelengths of radiation in driving a process to occur. So if you look here, you can see in the wavelengths of light, chlorophyll A, chlorophyll B play a role, as well as the carotenoids. In this, notice there's very little usage of green light here, and then you get some peaks over here. If you look at the profile of a plant light bulb, you'll actually see that it'll generally mimic this, where you'll have a significant amount of light produced in these wavelengths and very little here in the green wavelengths, so that way the plants can maximize the light that's provided to them and not get a lot of wasted light that's just going to be transmitted right through. Okay, so what this is taking a look at on the action spectrum is rate of photosynthesis measured by oxygen release. So as you can see, as you measure how much oxygen is produced, you get far more production over on this end and this end than you do in the center in the green wavelengths. Engelmann did an experiment over 100 years ago taking a look at this as well, utilizing algae. What he did was the action spectrum for photosynthesis was demonstrated by Engelmann 140 years ago almost now, 1880s. He exposed different segments of filamentous algae to different wavelengths of light. And areas receiving those wavelengths favorable to photosynthesis made a lot more oxygen, and those that were not favorable to photosynthesis, the wavelengths obviously produce significantly less, as you saw in the diagram. He used the growth of aerobic bacteria clustered along the algae as a determination of oxygen production, which is kind of ingenious. And again, a picture of the experiment. The action spectrum for photosynthesis is broader than the absorption spectrum of chlorophyll, and accessory pigments such as chlorophyll B broaden the spectrum used by photosynthesis. The difference in the absorption spectrum between chlorophyll A and B is due to a slight structural difference between the pigment molecules, as you'll see here. If you look at chlorophyll A at this location, you have a methyl unit, and if you look at chlorophyll B, you have a carbon, hydrogen, and oxygen located right in that position on the porphyrin ring. Okay, but structurally the rest of it is identical. Accessory pigments called carotenoids may broaden the spectrum of colors that drive photosynthesis. Some carotenoids function in photoprotection. They absorb excessive light that would damage your chlorophyll or react with oxygen. And when a pigment absorbs light, it goes from a ground state to an excited state, which is unstable. And when it falls back to the ground state, that excess energy is released as heat. In isolation, some pigments also emit light, which is an afterglow called fluorescence. So here you can see you strike a chlorophyll molecule with a photon of light. It will excite an electron and put it in what's considered the excited state. It's unstable here. For it to fall back down to its ground state, it's going to need to release all of that energy it picked up from that photon of light that brought it up here to begin with. So how it does that is it shoots it off as heat and photons of light, and sometimes we see that as fluorescence. Okay, a photosystem consists of a reaction center complex surrounded by light harvesting complexes. The reaction center complex is an association of proteins holding a special pair of chlorophyll A molecules and a primary electron acceptor. The light harvesting complex consists of pigment molecules bound to proteins, and the light harvesting complexes transfer the energy of photons to chlorophyll A molecules in the reaction center complex. These chlorophyll A molecules are special because they can transfer an excited electron to a different molecule, and that's really an important statement to remember. A primary electron acceptor in the reaction center accepts excited electrons, and it is reduced as a result. In solar power, transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of the light reactions. Okay, so here's the complex. You have a photon of light striking these receptors in the light harvesting complex, which is kind of this circular object right here. Of course, we're seeing a cross-section of it. And so the energy gets passed from one structure to another until eventually it gets passed to a special pair of chlorophyll A molecules. And then they will donate that electron of energy to the primary electron acceptor, which is all inside the reaction center complex. Now, this is the very first part of what happens. This is structurally what it looks like. You can see all the alpha helices and some beta pleated sheets in the structure, but this is what this purple piece looks like here. And you can see all the green chlorophyll pieces in there. Okay, there are two types of photosystems in the thylakoid membrane. There's photosystem 2, which functions first, even though it happens first. It's called photosystem 2 because it was first discovered. They discovered the second part, which they later named photosystem 1 afterwards. So at any rate, photosystem 2 occurs first, photosystem 1 follows. The reaction center chlorophyll A of PS2 is called P680 because it absorbs wavelengths of light at 680 nanometers, and photosystem 1 is P700 because of the wavelengths of 700 nanometers that it absorbs. Now, during the light reactions, there are two possible routes for electron flow. It could be cyclic or it could go linear. And linear electron flow, the primary pathway involves both photosystems. It produces ATP and NADPH using light energy. And there are 8 steps in the linear electron flow. First, the photon hits a pigment and a light harvesting complex of PS2, and its energy is passed among pigment molecules until it excites P680, and the excited electron from P680 is transferred to the primary electron acceptor. We now call it P680 plus. Okay, so that's going to all happen over, again, we're talking about all this happening over in this first section. So again, this is kind of that purple and green piece we saw a moment ago that was depicted in the cell membrane, but we have light hitting the green pigment molecules, passing the energy along until it hits those special chlorophyll A P680 molecules, which will donate the electron to the primary electron acceptor. In this process, water will also get drawn in. The electrons from, you get two electrons coming in from those hydrogen molecules, because as hydrogen becomes positive, it's becoming positive as a result of removing those two electrons. So these are the electrons from the hydrogen, as the hydrogen is becoming a hydrogen, just a proton literally, because there's no electron associated with it. And then the oxygen molecules are getting released, and of course you get one from each oxygen, so it's 1/2 of O2. Because remember, oxygen's diatomic, so it's always going to exist in nature as O2. So half of O2 is just an O. Okay, so that's what they're showing here. So this water molecule goes in. These are actually the electrons from the hydrogens, and the oxygen gets split off. Okay, eventually these oxygens, the 1/2 O2 will join up with another half O2 and form an oxygen molecule. By the way, those are the oxygen molecules that we're inhaling right now. Okay, then you go to the electron transport chain. Okay, you go through the cytochrome complex, which has a couple components to it. It's going to produce some ATP along the way, and then you're going to have some input into P700 here. Again, this is 700 nanometers of light, which is going to again generate an electron to the primary electron acceptor. This can also get light directly as well, because remember, these also are pigments, so they can absorb their own light, and it can go that way just as it does over here. Okay, so this happens as well as this. Okay, and then you get ferredoxin that's going to bring some of these electrons to NADP plus reductase, which will join up with NADP plus, reduce it to NADPH by adding that H plus in there. Okay, and that's kind of what's going on here. We'll take a look at all those steps a little bit more explicitly as we go through 3 through H, which we'll talk, 3 will actually pick up right here. So we talked about 1, 2, and then we'll go 3, 4, and so on and so forth. So 3, the water is split by enzymes. The electrons are transferred from the hydrogen atoms to P680 plus, reducing it to P680. It's the strongest, P680 plus is the strongest known biological oxidizing agent. The H plus are released into the thylakoid space, and oxygen is released as a byproduct of this reaction. At step 4, the electron falls down an electron transport chain from the primary electron acceptor of PS2 to PS1. Energy released by the fall drives the creation of a proton gradient across the thylakoid membrane, and that potential energy in step 5 will be stored, and the proton gradient will drive production of ATP by chemiosmosis, just like you saw in cellular respiration. And PS1, just like PS2, transferred light energy excites P700, which loses an electron to the primary electron acceptor. P700 plus is P700, which is missing an electron. So P700 plus will accept an electron passed down from PS2, photosystem 2, via the electron transport chain. That's happening in step 6. Now in step 7, each electron will fall down an electron transport chain from the primary electron acceptor of PS1 to the protein ferredoxin, which is represented by the FD. And then NADP plus reductase will catalyze the transfer of electrons to NADP plus, reducing it to NADPH. And the electrons of NADPH are available for the reactions of the Calvin cycle. This process also removes an H plus from the stroma. The energy changes of electrons during linear flow through the light reaction can be shown in a mechanical analogy. Kind of like you strike this little seesaw, which has an electron sitting on it, which will pop it up to photosystem 2, which will roll it down this little windmill, chemiosmosis, right? That's going to make ATP. The electron will keep going. Again, hit another kind of rest here. Another photon of light can be coming in at P700, right? Striking that. or the electron falls down, but either input is going to push that electron up into the primary electron acceptor of photosystem 1, which will then run through ferredoxin into NADPH and the production of NADPH from NADP plus via NADP reductase. In cyclic electron flow, electrons cycle back from ferredoxin to the PS1 reaction center via a plastocyanin molecule, that's the PC that you see in the diagram. Cyclic electron flow uses only photosystem 1 and produces ATP, but it does not produce NADPH, and no oxygen is released. Okay, so here's the cyclic. We looked at the linear. This was considered the linear. Okay, now from here, primary acceptor will donate back to ferredoxin. It'll get recycled through the cytochrome complex carried by plastocyanin back to the P700 plus unit, which will donate the electron back to the primary electron acceptor, and so on and so forth. Okay, so this is the cyclic version. Some organisms, such as purple sulfur bacteria, have PS1, but they do not have a PS2 system. And cyclic electron flow is thought to have evolved before linear electron flow. So, considered to be the older system, cyclic electron flow may protect cells from light-induced damage. Now, chloroplasts and mitochondria generate ATP by chemiosmosis, but use different sources of energy. Mitochondria transfer chemical energy from food to ATP, and chloroplasts transform light energy into the chemical energy of ATP. Spatial organization of chemiosmosis differs between chloroplasts and mitochondria, but also shows similarities. In mitochondria, protons are pumped to the intermembrane space and drive ATP synthesis as they diffuse back into the mitochondrial matrix. But in chloroplasts, protons would be pumped into the thylakoid space and will drive ATP synthesis as they diffuse back into the stroma. Make sure you understand the differences between these, as I believe there are questions that probably will show up on the test regarding the differences in ATP production between mitochondria and also that in chloroplasts. So, in how chemiosmosis differs. So I think that'll be helpful to make sure you understand those two sides real well. Okay, mitochondrion structure in chloroplasts. Here's another depiction of it visually of the differences between the two. You can see that there's a lot of similarities, and you can see the relationship between the structures of the mitochondria and what they're called in the chloroplast, but you can see that there's, here the thylakoid space, the intermembrane space, the matrix, the stroma. So there's complementary pieces really on each side that have a very similar process in how this occurs. So, at any rate, make sure you pay attention to that. Okay, ATP and NADPH are produced on the side facing the stroma where the Calvin cycle takes place. And in summary, light reactions generate ATP and increase the potential energy of electrons by moving them from water to NADPH. Okay, so we've talked about this light side here. We put water and light in. We get oxygen out, but we also get ADP in the inorganic phosphate made into ATP and NADP reduced into NADPH. Now we're going to use the ATP and NADPH that are produced in the light reaction to feed into the Calvin cycle. We're going to also take some carbon dioxide that's going to come in through the stomata, and we're going to carbon fix that in the first step of the Calvin cycle. And at the end, we'll actually halfway through the process, we'll actually produce some sugars, so carbohydrates. Okay, so here's photosystem 1, cytochrome complex, photosystem 2, which comes first, cytochrome complex, and then photosystem 1, ferredoxin, NADP reductase, making the NADPH, feeding into the Calvin cycle, chemiosmosis. So here is pretty much all of your photosystems, your light reaction, in great detail, visually depicted. This is kind of a nice diagram to keep in mind. Okay, let's take a look at the Calvin cycle. The Calvin cycle, like the citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. It's anabolic. It builds sugar from smaller molecules by using ATP and the reducing power of electrons carried by NADPH. So those inputs from the light reaction, ATP and the NADPH, are going to fuel the Calvin cycle. So carbon enters the cycle as CO2 and leaves as a sugar named G3p. You probably remember seeing glyceraldehyde 3-phosphate and cellular respiration. I think it was step 4 or 5 where it was produced in the energy investment phase. For net synthesis of 1G3p, the cycle must take place 3 times, fixing 3 molecules of CO2. Okay, so to make 1G3p, 3 CO2 molecules need to run through the cycle. Okay. The Calvin cycle has 3 phases: carbon fixation, which is catalyzed by Rubisco, reduction, and regeneration. So carbon fixation, reduction, regeneration. They're talking about one of the experiments they described. So here's your input of the 3 carbon dioxide entering the cycle simultaneously. So here's carbon fixation. You're going to put these together to make 3 phosphoglycerate. Oops. Okay. ATP is going to come in to make 1,3-bis phosphoglycerate. The NADPH will get utilized here in the reduction phase to make G3p. Okay, so you're going to have some intermediary steps here of phosphoglycerates of different types, 3-phosphoglycerate, because the phosphate's attached to the number 3. And then you have 1,3-bis phosphoglycerate because you have 2 phosphates now attached to the first and third carbon. And then here you have G3p, the phosphate again attached to third carbon, and then you have G3p coming out, and glucose and other organic molecules are coming out of this as the output here, G3p. Okay, and you have a continuation on to G3p, regeneration via ATP. ATP is going to donate the phosphate, so you can put 1 phosphate on each end. Okay, and so you can put 3 ATP in and get 3 ADP out. Remember, everything's triplicated. And then you'll have RUBP here, and then you'll go back to your starting point. Okay, dehydration, a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants close stomata, which are the pores, which conserve water, but it also limits the plant's ability to conduct photosynthesis. The closing of stomata reduces access to carbon dioxide and causes oxygen to build up. These conditions favor an apparently wasteful process called photorespiration. In most plants, C3 plants, where they have 3 carbons, initial fixation of CO2 via Rubisco forms 3-carbon compound, 3-phosphoglycerate. But in photorespiration, Rubisco adds oxygen instead of CO2 in the Calvin cycle, producing a 2-carbon compound. And photorespiration consumes oxygen and other organic fuel and releases carbon dioxide without producing any ATP or sugar, because it can't produce the right products. So photorespiration may be an evolutionary relic because Rubisco first evolved at a time when the atmosphere had far less oxygen and more carbon dioxide. Photorespiration limits damaging products of light reactions that build up in the absence of the Calvin cycle. And in many plants, photorespiration is a problem because on hot, dry days it can drain as much as 50% of the carbon fixed by the Calvin cycle. Now, C4 plants minimize the cost of photorespiration by incorporating CO2 into 4-carbon compounds. There are 2 distinct types of cells in the leaves of C4 plants: bundle sheath cells, which are arranged in tightly packed sheaths around the veins of the leaf, and mesophyll cells, which are loosely packed between the bundle sheath and the leaf surface. Sugar production in C4 plants occurs in 3 steps. The production of the 4-carbon precursors is catalyzed by Pep carboxylase in the mesophyll. And Pep carboxylase has a higher affinity for CO2 than Rubisco does. It can fix CO2 even when CO2 concentrations are low because of its greater affinity. These 4-carbon compounds are exported to bundle sheath cells, and within the bundle sheath cells they release CO2 that is used in the Calvin cycle. Okay, so here you got the mesophyll cells, bundle sheath cells. You have a vein, so this is the leaf anatomy. Here's your stoma, the opening. That's not right. That should be the stomata, I believe, right? The opening is the stomata, and the stroma is the fluid. I guess this is a singular stoma rather than plural stomata, maybe. I don't know. I just noticed that for the first time. Any rate, so here you have CO2, pep carboxylate. You have oxaloacetate. You probably remember this from cellular respiration, as well as malate in the citric acid cycle. So pep carboxylate will form oxaloacetate, which is a 4-carbon, malate, a 4-carbon. And then the CO2 will get broken off of that 4-carbon. So one of the carbons will get broken off into making carbon dioxide, and the other 3 will remain together as pyruvate. Again, another relic from cellular respiration. And the pyruvate will then go through some effect from ATP, which will get broken down, phosphorylated, and the ATP will break down into ADP, and you'll form PEP, which is again a 3-carbon version, which will pick up the 4th carbon from the incoming CO2, making it back via pep carboxylate into oxaloacetate, a 4-carbon item again, and around it goes. Now this CO2 comes off here in the bundle sheath cell, and it goes to the Calvin cycle, produces sugar, and so on. So the big difference here is that you have this additional step here in a C4 pathway. Okay. Since the Industrial Revolution in the 1800s, CO2 levels have risen greatly. Increasing levels of CO2 may affect C3 and C4 plants differently, perhaps changing the relative abundance of these species. The effect of such changes are unpredictable and a cause for concern. Suitable agricultural land is decreasing due to the effects of climate change, while the world demand for food continues to increase. C4 photosynthesis uses less water and resources than C3 photosynthesis. Scientists have genetically modified rice, which is a C3 plant, to carry out C4 photosynthesis. They estimate 30 to 50% increase in yield compared to C3 rice. Some plants, including succulents, use crustaceous acid metabolism, CAM, to fix carbon. CAM plants open their stomata at night, incorporating CO2 into the organic acids that are stored in the vacuoles. And then they close during the day, and CO2 is released from organic acids and used in the Calvin cycle. The CAM pathway is similar to the C4 pathway in that they both incorporate CO2 into organic intermediates before it enters the Calvin cycle. The C4 pathway structurally separates the initial steps of carbon fixation from the Calvin cycle, and the CAM pathway, these steps occur in the same cell, but are not separated by time like they are in the C4. Okay, so here's an example, C4, sugarcane, and on the right, pineapple, CAM. Okay, you can see here the process is virtually the same, except in A there you have a separation spatially between 2 different cell types. On the right, it's all occurring in the same cell type, but you have a temporal or time difference, a time separation between night and day. So kind of interesting. Difference between C4 and CAM. Okay, the energy entering chloroplasts as sunlight gets stored as chemical energy in organic compounds. Sugar made in the chloroplast supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. Plants store excess sugar as starch in chloroplasts and other structures such as roots, tubers, seeds, and fruits. Okay, so here's kind of the whole picture now. We took a look at the light reactions with Photosystems 2 and 1. The production of ATP and NADPH, which feed the Calvin cycle, with carbon fixation of CO2 onto 3-phosphoglycerate, making G3p and sugars ultimately, and then getting returned back up to be recycled again. Again, leaving off ADP and NADP plus to be used by the light reaction again, and so this whole system goes round and round. Okay. Again, tying everything together, we talked a lot about the nucleus way back when and the production of DNA into messenger RNA, traveling through the nuclear pore, forming a protein and a ribosome, right, as the messenger RNA gets read and transcripted and translated into that protein, where it's then going through modifications through the rough endoplasmic reticulum or the Golgi apparatus, and then it gets, when it's finally processed, it goes out into the cell or into your system, and it does something, right? So Now taking a look at this new information we put together in this chapter on photosynthesis, you can see the effect of in a plant cell chloroplasts in photosystems 1 and 2, and how they interact with mitochondria and the ATP, CO2, H2O exchanges that occur between all of those. All right, so that's kind of it for chapter 10. And again, good luck with this, and ask if you have any questions. Thanks. Bye-bye.